Kendra is another extended, like the family member that came in as part of what is now kind of a multi-institutional affiliation. So she has a history with Tulane University, and then went meandering a bit and then came back. But her role is now that she runs radiation oncology for our entire health system. So her influence in our being able to achieve things beyond East Jefferson, I think, is going to be great. And then we and she have this passion for bringing the radioligands to this part of the country. And then one other little push for clinical trials, as I've told many of you when we've discussed them, is that it really is about meaningful. They make sense in this part of the country, and it's bringing access to a part of the country that I'll tell you is generally left behind when it comes to access to those type of things. And so I think we're really kind of getting our act together, and maybe we just learned a lot of stuff from the pandemic. We had a lot of time to think about what we needed to do.
Please welcome Dr. Kendra Harris.
Hi, y'all. So as was mentioned, I am a radiation oncologist, and so today's talk is just going to focus on some of the ways in which, like, my expertise could lead to a little bit more concrete understanding of basically radiopharmaceutical deployment for neuroendocrine cancers. And I always like to start with this lady. I don't know if anyone knows who she is. This is Marie Curie. She is the woman who actually identified the first radioactive element. She also is the only human ever to be awarded 2 Nobel Prizes, one in physics and one in chemistry. So I always give a little nod to her. So basically today I'm going to focus on Theranostics generally, and then the clinical context for our discussion. I'm going to talk a little bit more about the landscape of molecularly directed radiotherapy overall, nerd out a little bit on you for some alpha and beta talk, and then return back to some practical considerations. All right, so you have probably heard this already today, but generally Theranostics, as a big umbrella, is the idea that you would basically use a particular targeting molecule, which we're going to refer to as the ligand, to identify the location of particular tumors using PET scans. And then you're going to pop the thing attached to the ligand off, which has allowed you to see it, and instead attach a therapeutic radio particle and re-administer therapeutic radiation connected to this targeting molecule, so that the same thing that you were able to see using the PET scan will, at the time of dosing, receive therapeutic radiation. So in this example, right, what we see is this is a cancer cell. For today, we're going to think about this as the somatostatin receptor that is present. This set of, shapes represents the thing that recognizes the somatostatin receptor, which for neuroendocrine cancers is a somatostatin protein complex, and it can be connected through a linker to a radioactive compound. And that compound, when you're doing imaging, can be like copper for a copper PET scan or gallium for a gallium PET scan. And when you're delivering actual therapy, instead of the PET radioisotopes, you're attaching something that will deliver treatment. So for the Lutathera, that's lutetium. For some of the newer trials, that would be like lead. And so in the context of today's discussion, I love this little cute little GI tract cell here. The purpose is that our focus is on this somatostatin receptor. And We now have a roughly, I guess, 7-year history of FDA approval for adults of using a recognition of this receptor attached to a radioactive element as Lutathera. Just a month ago, this treatment was expanded and is now approved for the treatment of children with neuroendocrine cancers. In fact, it's the first FDA approval ever for molecularly targeted radiation for minors. And then right now, what is approved is lutetium, but as we're going to talk about, there's actually a whole bunch of different things that you could connect to the somatostatin receptor, and indeed a whole range of targeting molecules that are being evaluated for deployment, not just for neuroendocrine cancers, but across a variety of cancers. Okay, so this is a question I get asked a lot, which is, how is it that you can give radiation but not cause all the normal cells to die? And I find that a goat in a beret is highly memorable. And so this takes us back to one of the critical observations that allowed the start of what I'll call empirical radiation oncology, which was just the observation that if you had a male ram, I guess that's redundant, but in any event, if you had a ram that you wanted to sterilize and you took a piece of radioactive rock and you held it against the testicles for an hour, you would have a sterilized ram, but about 2 weeks later you would have a dead ram because the skin would die and they would develop a terrible infection. If instead you took that same radioactive rock and you held it to the testicles for 5 minutes, 3 days in a row, you'd have a frolicking ram who was also sterile. And so was the first observation of some relationship to how radiation is delivered and the ability of normal cells to recover. And that there is a difference in the ability of cells generally to recover. We now know that the way that radiation impacts cancer cells is that it causes breaks in the blueprints of the cells. So within the blueprints of the cells, you have all this DNA. The DNA is what we use predominantly when do we use our blueprints? Well, when we're building, right? So the main time that all of our DNA is used is when a cell tries to divide. And so when a cell is dividing, what it has to do first is make a copy of its blueprints, and then it goes ahead and divides. During that copying process, if there is a problem that breaks one side or both sides of the backbone of the DNA, what can happen is that the replication process sort of gets stuck where that break happens. Now, you would imagine that, hey, being able to fix your blueprints is kind of important. And in fact, all of our normal cells have at least two major ways to repair that kind of damage. Single-stranded repair and double-stranded repair. So there's a fancy name for those things, but in any event, this is why if you get a bad sunburn or you live in New Orleans or you have some other type of exposure, you don't just have your skin peel off, because your normal cells have the capacity to repair many types of this kind of blueprint damage, but it takes a little bit of time. By contrast, cancer cells usually get rid of things that help repair DNA damage, in part because all cancers are the result of an accumulation of errors. And if you get the right set of accumulated errors, you can end up with neuroendocrine cancers or other types of cancers. And one of the ways that that occurs is that they lose ways to repair mutations, and just by chance, you can end up with a mutation that causes a cancer. And in this way, there is a big difference between how lethal radiation is to cancer cells and how lethal radiation is to normal cells, so long as you take precautions. And one precaution is that it makes sense to deliver radiation. Maybe you want to give a certain amount, but instead of delivering that amount all at once, you give it in smaller doses. And that interrupted dose approach is really about letting your normal tissues that are impacted by the radiation have time to recover. And so you could imagine that if you gave a dose of treatment, it would impact both your cancer cells and your normal cells. And then you're given a treatment break and your normal cells go through this recovery process, but the cancer cells can't because they jettisoned the equipment they need to repair that type of damage. And then you give another dose and both are impacted and your normal cells are allowed to recover, and you get this sort of separation of how lethal radiation is for normal cells and how lethal radiation is for cancer cells. Getting even a little more nitty-gritty, it turns out that when we think about radiation generally, there are a bunch of different kinds of radiation. And for example, when patients come to my clinic and get treatments Monday to Friday and it's non-invasive and they lay there and they can't feel anything but they're receiving treatment, that's external radiation treatment. That is delivered with a type of radiation called photons, which are like waves and particles, both neither, very kind of a wily situation there. But those photons and all radiation can have both direct effects where the radiation element or the energy packet, we'll call it, can actually directly hit the backbone and break it. But actually what is much more likely to occur is that the packet of energy interacts with water, and the water is split and you get a free radical, and it's the free radical that interacts with the backbone to break it. So a direct effect would be, gosh, that packet of energy that's passing through directly impacts it, but much more common is that whatever the packet of energy is interacts with water and breaks it. And you can have both single-stranded and double-stranded events. So that's one kind of radiation, would be this external radiation. But today we're talking about theranostics, and we're talking about the theranostic that has been approved, which is the somatostatin conjugated lutetium. And in that circumstance, the lutetium is what we call a beta emitter. And we're also going to be talking about alpha emitters. And basically, beta emitters and alpha emitters are just two different flavors of radiation. They are not like the external beam radiation. But as I'll talk about in a couple of minutes, these different flavors, they have their effect in the same way. Whoa. All right. Yeah, so this is what we're talking about, is this Lutathera. Basically, it gives off electrons, and these electrons are what have the effect on the cancer cells and can have an effect on the normal cells that are very close by. But as we talked about, this radioactive element Lutathera is connected to the somatostatin receptor. So that receptor is put into the bloodstream. It finds its, so you sort of think about it like a GPS system, looking for those somatostatin receptor expression on the cancer cells. And then, having been brought very close to where the cancer cells are, it has drug along this radioactive element, which is kind of pew, 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 giving off electrons, which don't travel very far, but can cause that DNA damage to the cell that's closest, which we hope for the most part is cancer cells. And so this is, in practice, what many of you have experienced, which is that you are sent for a type of PET scan. This was the picture I could find, but I think we're kind of partial to copper PETs around here, which allows you to see where the spots are that the cancer is predominating. And then you give a treatment with the same targeting molecule attached to lutetium. And then you take another PET to evaluate what residual expression you can see of that somatostatin receptor, which we take as a proxy for the disease itself. And now we nerd out. So I'll just tell you that I wandered around the radiation oncology department, I would say at least 12 months before I understood even the language that was being used, having had 10 years of postgraduate education. So beta therapy, lutetium. I'm going to talk about these two general categories of radioactivity that we're using in Theranostics by comparing them to one another. And in general, this beta therapy group, of which the lutetium is a member, behaves more like other types of radiation that we use in terms of how effectively it kills cancer cells. In general, it has a little bit longer distance over which it can act. In general, it causes less damage. In general, we worry a little bit more about radiation exposure because that particular element has some daughters. I don't know why they're always daughters instead of sons when it comes to emissions, but in any event, it has some emissions that are able to move or be detected far enough from the person that has received this radiation that we worry about, exposure a little bit more. Alpha therapy is, for example, what the elements lead and actinium are known for doing. The thing about alpha therapy that I think about is like the electron is this itty bitty particle and it has a sort of itty bitty effect, whereas alpha emissions involve the expulsion of larger particles. And so in general, alpha emitters and alpha emission is something that causes more damage per emission. Although most of our original understanding of this comes from basically irradiating bacteria and asking how well alpha emitters kill bacteria as opposed to how well beta emitters kill bacteria. But it is recognized and referred to as a linear energy transfer, sort of this idea that per punch there's more force, it causes more trouble. The other thing that is true is that the action takes place over a shorter distance, which I think about as like they're heavier. I had the misfortune during a clinical trial I was participating in where I was personally drawing the PK blood draw. So I had administered an alpha emitter and I was drawing the blood up that was filled with this radioactive substance that the patient had just received. And I managed to spray that patient's blood all over myself. And I had an opportunity really to, test what I've been told, which is that with alpha emitters, the radiation moves such a small distance that it really doesn't even make it out of the liquid it's contained in, that a sheet of paper will block the alpha emitter. And I can tell you that that's true. So that is sort of the lutetium and these newer elements that we're seeing make their way into neuroendocrine treatments. But when it comes to understanding the potential that Theranostics offers, I like this slide, which is that we're talking about this guy right there, and then I'm going to touch on this lead. But actually, there's a whole bunch of these, and there have been deployments of a variety of these already in cancer. So radium-223 was the first FDA-approved anti-cancer therapeutic that was an alpha emitter. Lutetium is approved by the FDA both in neuroendocrine cancers and in prostate cancers. And there is quite a substantial amount of activity right now in terms of looking at different combinations of radioactive elements and how to best target those elements. One of the most exciting alpha emitters making its way through neuroendocrine clinical trials is lead. Now, it doesn't sound like a great idea to just inject lead into your bloodstream, but actually the total concentration needed is very small. And again, this lead is connected to the receptor that's going to preferentially carry it to your cancer cells. One of the reasons that this particular element is attractive is that there's sort of like a Goldilocks problem when you think about putting something in the bloodstream that's going to attach to cancer cells and deliver radiation. And that is, you want to have a targeting molecule that will bind to the target of interest for just the right amount of time. And that during that just the right amount of time, you want to have enough radiation emissions for that binding to result in cancer cell damage. And there are some qualities of lead that suggest that it's a good candidate in this regard. And in fact, was recognized about 90 days ago in neuroendocrine cancers by the FDA as sort of a breakthrough designation. The FDA has increasingly, to their credit, tried to be sensitive to the idea that when we have very promising therapies as people are waiting for them, it can take a long time for those therapies to reach approval. And so when such really promising therapies are developed, they're trying to shunt them into a somewhat expedited FDA approval pathway, of which this lead dotamate was recently placed into. When it comes to emerging alpha therapies and just theranostics in general, I think the message that I want to communicate is how exciting this area is in terms of what is going to be coming down the pike in the next 3 to 5 years. Certainly the lead directed neuroendocrine trials are ongoing, and they're soon to open sort of the critical phase 3 randomized look at the deployment of somatostatin conjugated lead in neuroendocrine patients. But that is not the only thing that is being looked into. And in fact, when it comes to all of nuclear medicine, if we look at like the entire use of nuclear materials in a medical context, which is a combination of things that we do to take pictures of people and things that we do to treat people. This year, of that pie, about 20% of the pie is being deployed to treat people with therapeutic molecular radiotherapy, like Lutathera. And it is predicted that by 2026, that market will be almost 50-50. So the amount of deployed therapeutic radiation that is liquid will go from 20% to half. and by 2031, 3/4 of all radioactive elements used in the United States will be used in order to treat patients' cancers. Which is to say that the clinical trial deployment activity as of this year, which is represented here, I think, like, given the fact that the zebra is the appropriate animal because of the need for awareness that even rare things happen often enough that there needs to be an awareness to look. Despite the relative rarity of neuroendocrine cancers overall, it is the second most important nexus of clinical trial activity for Theranostics. And one of the places where I think there's a real chance that the next FDA-approved alpha particle will be to treat neuroendocrine cancer, and that is pretty awesome. It's also true that there's 83% of other things being looked at. And in Theranostics, that is the see it, treat it paradigm, some of the investigations and some of what is learned in neuroendocrine trials is very specific to neuroendocrine cancer, but some of it is not very specific. And so there is a synergy that happens when there is more attention overall paid to this approach to managing and treating cancers that I'm excited about personally. Before I joined the team at East Jefferson, my primary source of experience actually was in prostate cancer. And the sort of sentinel event in prostate cancer was the approval of radioactive radium, which was not molecularly directed, but was the first FDA approval of an alpha emitter. And what followed in the nearly 20 years since that was this crazy explosion, both in terms of identifying new ways to target prostate cancer cells, and then getting more and more and more ambitious with what types of radioactivity could be attached to those targets. And really, in terms of having a lot of different ways to target neuroendocrine cancers, this is a place where there is, I think, opportunity. With 3 minutes left, what I think is really important is, so this is my nearly 20th year since graduating medical school, and I have worked at a variety of places, some of which were more aggressive about clinical trial enrollment and some of which were less aggressive about clinical trial enrollment. And I will say that from my point of view, up until I had involvement with Theranostics clinical trials, I just hadn't had contact with clinical trials where the benefits that could accrue to the patients I took care of were so clear to me as to cause me to, I don't know, become a rabid dog when it came to certain types of advocacy. It seemed to me actually generally that many of the clinical trials that I had exposure to in my early career were at least as likely to cause harm as good. And I had never enrolled patients in a trial in which, like, I could see the impact in an unassailable way. And then I got involved in Theranostics trials, as I said, initially with prostate cancer. And I had the lived experience of watching people who were suffering terribly, who had a lot of very serious consequences of where their tumors were, and then had them enroll at that time in a randomized trial that was balanced 2-to-1 to try to make up for the fact that 1 out of 3 people wasn't going to get this medicine. And just watch it really change the lives of the patients and the families that participated. It's part of what made me really excited to learn about the neuroendocrine group's work at East Jefferson and actually across time at several locations. Having had the contact so far with the trials that are open, it's a similar experience. And it's one of the experiences of really feeling the urgency of what it means either to have access to new agents through that mechanism or not. And so I'm proud to be riding the coattails of these folks over here in terms of participating and contributing, because actually deploying these types of agents and running clinical trials that deploy these types of agents is a pretty difficult undertaking. And I've just listed here kind of some of the special needs that I have observed as arising. And that's it. Thank you.